So the conclusion about mercury is that the children with ASD do not have higher levels of blood mercury, but I do need to point out that this was not a test of the hypothesis of whether mercury causes autism. For that, we would need to look at mercury at a much earlier point in time, and we are, in fact, getting ready to analyze data on hair mercury and blood spots. The California newborn screening program saves a little drop of blood for every birth in the state, and it's stored in a biobank which researchers can have access to. Okay, let me talk a little bit about air pollution. We were interested in whether the residences of the mothers at the time of delivery might be closer to traffic and other sources of air, specifically traffic sources of air pollution, as compared to the children who develop typically. So we used their maternal address at the time of birth, and we also looked at residences they gave for the pregnancy period and compared them. We mapped those to maps that had all of the freeways and major roadways. What we found was that there was a higher prevalence of mothers who had lived close to the freeway in the closest 10% within 300 meters, with an odds ratio, meaning the odds that their child would develop autism was about almost 1.9-fold higher compared to the non-cases. So this correlates very well with exposures, because at about 300 meters is when there's a big drop-off in pollutant levels in ambient air. We also know that the components of traffic-related air pollution, things like benzoate-pyrene and ozone, are known to induce developmental deficits in rodents. Also, PAHs, polycyclic aromatic hydrocarbons, can cause oxidative stress and inflammation. So these are all plausible mechanisms that could be related to development of autism. Now I'm going to turn to the immune connection. We work with a number of laboratories, as I mentioned. Dr. Judy Vandewater and Paul Ashwood conduct immunology studies on the blood samples from the children. And what we have found, and this is a smattering of some of our papers that we've published, that the children with autism do seem to have some dysregulation of their immune systems. They respond very differently to antigens. They have different levels of immunoglobulin G. Some of the cytokines, which are chemicals that are the messenger molecules from lymphocytes going out to help the body fight infections or fight foreign bodies that enter, really are different between the two groups. And then this is a very striking finding that we've published and we've also followed up on now. Mothers of children who have autism are more likely to mount a response, a reaction to fetal brain tissue. So this is in the laboratory. This is not, we're not looking in the body. We draw the blood. We challenge with, we expose the cells to this fetal brain tissue, and we see that those mothers who have a child with autism might make antibodies to this fetal brain tissue, but none of the mothers of typically developing children do. In our first study, we found it was about 12% of the mothers with children with ASD. In later studies, it's maybe somewhere between 6 and 12%. This initial paper was about 60. We now have looked at hundreds of cases and hundreds of controls, and the finding is holding up. Now, what does it mean is not entirely clear. It's possible that something immunologic goes on, creating an inflammatory environment in the brain that could lead to the development of autism. It could be the other way around, that the neuropathology influences the development of the immune system. Or it could be neither of those. But we do think that there's something going on here that really needs to be understood. And it tells us that it's not just a disorder of the central nervous system. There are other systems involved. And one of those other systems really has to do with very basic cell function, which is mitochondria. The mitochondria are the battery of the cell. They're the energy production. And they're particularly important in brain cells. And what we found in this very recent paper is that several markers of mitochondrial dysfunction were more extreme in the children with autism. In one case, plasma pyruvate which was seen to be outside the range of the controls in 8 out of 10 of the autism cases. And we did not see lactate being different, and therefore the lactate to pyruvate ratio, which is a very strong indicator of an imbalance, was lower. Also, higher rates of hydrogen peroxide production. Hydrogen peroxide is an indicator of stress in the body. The epidemiology here has a kind of a relationship with the bench science. So those are some of the bench scientists we work with, immunologists and molecular biologists. And we also have a relationship to social policy, that the findings that come out of epidemiology can hopefully provide us with guidance for ways to actually intervene and modify the risk of autism. Because what I want to see is those rates starting to come down. So I'm going to finish up with talking a little bit about a couple of other environmental factors that we have some data on. PBDEs. These are polybrominated diphenyl ethers, which are flame retardants, and they're used widely in consumer products. In Sweden, they started noticing the levels of PBDEs going up in the 1990s, and so they issued a ban, and then the European Union followed, and then it took another 5 or 6 years, and the US did something sort of like that. We banned most of the PBDEs, but not all of them. Meanwhile, in California, we've been increasing the use of PBDEs because the chemical industry has lobbied ferociously to make more stringent flammability standards. And so they are in a variety of products that are listed on this slide. But they interfere with thyroid hormones, which are really critical to prenatal development, sex steroids, and there are animal and actually now some human studies suggesting neurodevelopmental deficits related to PBDEs. So we looked in a pilot study, very small number of cases and controls, to see whether the PBDE levels would differ between the cases and the controls. And our thinking at the time was that PBDEs are very persistent and that we could look at the level in the 2 to 5-year-old and maybe it would give us a clue about what the exposures might have been in the first year of life or possibly even prenatally. And we were also interested in what factors contributed to the PBDE levels. And what we found, is shown in this slide, is that some of the big contributors to PBDE levels were consumption of chicken, pork, purchase of a new mattress, and other foam products in the house. Now, this actually turned things around a little bit because people thought that most of the exposure was volatilization from these household products, just it gets into the air. But this showed that actually the PBDEs have gotten into the food chain. Which is what happened with PCBs about a generation ago. That first it's in the household products, and next thing you know, it's in the environment, and it's worked its way up into the food stream. So we also found, as I said, California has high levels. We found levels of PBDEs that were higher in these children than in any other previously reported population. But we did not see a difference between the ASD children and the typically developing children. We don't think this means that these are necessarily safe in terms of ASD. We don't think this is the last word because of this finding about the food chain, which we did not know about when we started the study. It suggests that because diet changes dramatically between birth and by the time the child is 2 to 5 years of age, that what we measured was probably not a good indicator of their very early life exposures. So we think that more work needs to be done looking at measures of exposure earlier in time, and we've gotten a little bit of supplemental funding for our grant to be able to go into the homes and collect dust samples using a HEPA. It's a very high-efficiency vacuum cleaner that digs out the deep dust, which can actually reside for years in the carpets. So the significance of the CHARGE study, I think, is that it really is the first comprehensive study of environmental factors. I'm going to tell you about a couple of things which I don't have slides on, but you'll be hearing about this finding very soon, and the paper has now been accepted, so I think it'll be coming out within a month or so. One of my postdocs came in and decided to look at nutritional factors, and she went through the questionnaire and pulled together information on whether mothers had taken prenatal supplements and when they took them. And she found that the mothers who took prenatal vitamins in the 3 months prior to pregnancy and the first month of pregnancy had half the risk of delivering a child who developed autism. This is really significant because this is something that is not that difficult to do. It's difficult at the population level because most pregnancies do not seem to be planned. But it is a way in which people can actually intervene if you are planning a pregnancy by taking those prenatal vitamins before you get pregnant. It's interesting because this is the same time period that we recommend women take these anyway because it reduces the risk for a neural tube defect, one of the most serious congenital malformations. So it may be that there's something about that early development of the neural tube, the beginning of central nervous system development, that is particularly vulnerable to what is in prenatal supplements. And the prenatal supplements are different from multivitamins in that they have a much higher level of folate and they have a much higher level of iron. So we're going to be looking a little bit more at what the individual components of this might be. But I think it's going to really, it says that there are things that people may be able to do to reduce their risks. The other thing about this finding is that we see a gene-environment interaction. So when we look at some of the genes that metabolize folate, we see that certain genes are associated with a much higher difference between those who do and don't take the prenatal supplements. So it may be that there are some individuals who are particularly susceptible and really need either the higher levels of folate or iron. Now, the limitation in the CHARGE study is that it's all retrospective. But what people remember, of course, can be faulty. So even this study, I think it does need to be replicated. We need to be looking at it in a situation where we're not asking the mothers to tell us what they took and when they took it, but it's done before they know the outcome of their child. Nevertheless, this is really how the study is going about, trying to find the ways that we can intervene to bring the rates down. To overcome this limitation of retrospective data collection, we also launched the MARBLE study, Markers of Autism Risk in Babies Learning Early Signs. And this is a study of pregnant moms who already have a child with autism. We bring them in, some of them pre-pregnancy even, early pregnancy, and we're collecting information and specimens. Some of those children will develop autism, probably somewhere about 10 to 20% of this subsequent pregnancy. The others will not, and we'll make that comparison. And we'll have all of the data prospectively in real time. Another study of the same design is called the EARLY study, and both of these studies, they complement the case control approach. They eliminate the problem of this differential reporting because you already know and you've been thinking really hard what went wrong with this pregnancy. It permits the examination of temporally relevant exposures, and of course the control mothers maybe haven't spent a lot of time worrying about what did they do wrong. So that's where that difference comes in. But these younger SIB cohorts are really focused on the multiplex cases, right? So if there's a different set of environmental factors that influence cases that, are these family-based cases, then the broader range of cases, we might not find it in the MARBLEs and the EARLY studies. And that's where the National Children's Study comes in, because that's where we'll be looking at way more of these simplex cases where it's just one child in the family. I just have to acknowledge the large number of investigators who have contributed to all of this work. It's been a huge study. We had people all over Davis and many other parts of the state and the country and the world working with us on these various projects. I also need to acknowledge a huge number of people. This is just a small part of the staffs of our studies. The top two people have been data analysts who worked on the PBDE and the trend analyses. And then many, many families. We can't do these studies without the families. And we've had about 1,500 families in CHARGE, over 200 in MARBLEs. And then, of course, our funders, without whom we could not do any of this research. But funding is always tenuous, and we are actually coming up for renewal on several of these studies, so we're going to be launching a big fundraising campaign to make sure that these studies keep going. So thank you very much for your attention.